Hello, my name is Michael Fleisher, and today I'm going to be presenting my research, which focuses on Aza-Peptide carbonyls, which we believe will be an effective new class of proteasome inhibitors. And an inhibitor does exactly as it sounds, it just inhibits or stops something. And the thing that it's going to be inhibiting in this case is the proteasome. And the proteasome is just a large protein complex that plays an integral part in the ubiquitin proteasome pathway. And this pathway is found in almost all cells, and it's just responsible for protein degradation, so basically recycling building blocks within cells. It's responsible for immune response and apoptosis, which is just cell death. And this is important because it's been found that proteasome activity has actually increased in certain cancer cells. So this makes it a promising target to treat such cancers, particularly in this case, multiple myeloma. So the purpose of this research is to target the immunoproteasome for the treatment of autoimmune diseases and inflammatory disorders. So if you look at the first picture under the introduction section, you'll see how the immunoproteasome differs from the constitutive proteasome. And it's just by three beta units, particularly beta 1, 2, and 5. And this is important when thinking about the specificity of the proposed inhibitors. So if you'll look to the proteasome nomenclature and current inhibitor sections, you'll notice that there are actually a couple proteasome inhibitors already on the market. And these inhibitors all are going to consist of a peptide sequence, which is just a bunch of amino acids in a specific order, and then a chemical warhead attached to the very end of those. And this is the part of the inhibitor that does the inhibiting, effectively shutting down the proteasome. And the two that are on the market are the most widely used for treatment of some cancers are carfilzomib and bortezomib. The only issue with these is that they've been linked to particularly bad side effects, bortezomib being peripheral neuropathy. Now, if you look to the design section, you'll see that our inhibitor features an alpha carbonyl hydrazide as well as a chiral P1 Aza amino acid residue with an optimized peptide portion to the preferred proteasome substrates. And this basically all just means, to sum it up, is that we've chosen specific sequences of amino acids and chemical warheads which will help limit off-target activity. So in terms of how these inhibitors are achieved, like within the lab setting, if you look at the synthesis section, this is predominantly what I've been working on. And this section will take you just step by step through the process. And each of those will be an individual reaction that I will set up and I'll work up and all of that stuff. But first I'll start by doing a coupling, which just means I take two different amino acids and I'll couple them together. And then that'll be the start of our peptide sequence. And then I'll just do as many couplings as is needed for whatever sequence we're trying to get. And then once that sequence is done, we'll do a chemical warhead coupling. And then the result will be a completed inhibitor, at which point we'll go to testing. So these finalized inhibitors will then go on to be tested to see how effective they are at inhibiting the constitutive or immunoproteasome. And you can see these rates of inhibition on the inhibition data table. And the ND on this table just means we haven't tested the inhibitor yet, but we will as soon as we can. And then in conclusion, we have synthesized the first Aza-Peptide carbonyl inhibitor to target the immunoproteasome with a noroleucine at the P1 residue, and we're in the process of optimizing the peptide portion to obtain a immunoproteasome specific inhibitor. And we'd like to acknowledge the Ohio State University Technology Commercialization Office Accelerator Award, as well as all the wonderful references below for the excellent representations. Thank you.